This video is on the replication of DNA. One of the beautiful things about the Watson and Crick model for DNA is that it provided a method for duplication of DNA within the cell. DNA to DNA is called replication. If this is the parent DNA molecule represented by this double helical molecule, we believe that the replication is semi-conservative. That is to say that the two daughter DNA molecules produced each have one strand of parent, the black strand, and one strand of newly synthesized DNA, the blue strand. Two identical daughter DNA molecules. Let's look at the process in a little more depth. Here we've got a hypothetical sequence on a DNA molecule, two complementary strands. Opposite every A is a T, opposite every G is a C, C and G. We believe that the first step in replication involves a separation or unwinding of the double helix. This involves breaking the hydrogen bonds between those base pairs, and by separating the two strands now, it allows new nucleotides to align themselves and to form new hydrogen bonds. Each of these strands serves as a template for the newly synthesized strands. If we show these newly synthesized strands in blue, again, complementary base pairs would form. Opposite every A is a T, opposite every G is a C. And notice now that these two daughter DNA molecules that are produced are exactly identical, and they're identical to the original parent. Let's go to the third board. Here's another view of replication and process. Parent strand over here, as it starts to separate, it exposes these base pairs. And remember that the double helical DNA molecule is anti-parallel. That is to say, this strand has a 3' end over here. It's running just in the opposite direction. This would be the 5' end of the molecule. And it's a little bit like pulling a rope apart, a twisted rope. And as we unwind the rope and expose these base pairs, this point is called the replication fork. It's an enzyme catalyzed process. It would take energy to break those hydrogen bonds, and to assist in the process, making it very efficient, is the enzyme helicase. Let's look at the next board. Okay. DNA polymerase catalyzes the synthesis of the newly synthesized strands. And we believe that the daughter DNA molecules, or I should say the newly synthesized strands, are formed in the 5' to 3' direction. So it would align itself in an anti-parallel fashion over here. This is the 3' end, and it would proceed down, and as it does so, nucleotides are linked together to form one continuous strand. Over here on this side, however, because it's running in the opposite direction, it means that DNA polymerase is going to have to jump over to this side and synthesize a portion of DNA as it's exposed right here. Each time it jumps over, it produces a short segment, and each of these segments have a gap between them. That's called the nick. And the discovery of these fragments over here on this side was done by a man by the name of Okazaki. So they're called Okazaki fragments. So one strand is synthesized completely. Over here, the other strand is synthesized in fragments. It's necessary to seal those together, and that's catalyzed by the enzyme DNA ligase. Incredible that nature could come up with this method for replication of DNA. It's based upon the four letters A, T, G, and C. Whereas in digital work these days, we use a 2-letter code. Nature came up with its own way of passing information along.